side. It's going to be one of those days. That makes sense? Different distribution of these soil nutrients forms different distributions of the plant species, but there is this kind of overlap as this concentration tapers off a little more gradually. Okay? All right, so far so good. Ecologists, depending on which type of community they work in, if a community ecologist works in an open community, the ecologist is thinking very differently about communities. They don't have these defined barriers. Species drop in and out of the community all the time based on where you are along the environmental gradient. On the other hand, individuals that work in these closed communities, for them, there are very, very clear boundaries between communities. These ecotones are very easy to pick out. The ecologists that first started to work in communities pretty much worked in these closed communities. So the way we think about communities today tends to be defined by work in a lot of closed communities. That is, communities have an identity. It's very easy to determine where the community starts and where it ends. And then we also think a lot about the attributes associated with these closed communities, right? I'm calling these community parameters there. They're parameters, they're attributes, they're characteristics that we can use to define this community, okay? So I have a couple up here. We're going to talk through these a little bit today. The first attribute of the community we'll call species richness. This is defined as the number of species. Again, that's the plural, right? SPP is plural, SP period is singular. The number of species in the community, that's called the species richness. Now you should be thinking in your head, okay, well, why is that a closed community characteristic? Well, if it were an open community whose distributions of species are coming in and out all the time, right? Some species with broad distributions, some of them with narrow distributions, but I can't predict where along this gradient any species will be by looking at other species. Well, that means if I'm standing here, there's a certain number of species, but if I'm standing here in the same community, there's another number of species, and so on. Whereas if I can define out this closed community based on the presence of this ecotone, I'm defining the community here, I get one number that measures the number of species, whereas here it really depends on where you are along the environmental gradient. Okay, all right, number of species. That's species richness. Then we have some abundance measures. Later today, I'm going to break these down mathematically for you. But for right now, there are three measures. Density of species, that's the number, density of a particular species, that's the number of individuals of species X divided by the area that I looked at, and that's number of individuals per area. That's a density measure. For some species, it probably doesn't make sense to think about them in terms of density, and that's because individuals can vary a lot in size. So for something like a tree, a coral, or a clonal plant, remember doing those when we were talking about reproduction, where whole mountainsides could be one aspen tree in Colorado. For these, it may make more sense to think about the size of individuals. So this is area taken up by our species divided by the, well, no, it's just an absolute measure. So it's the area taken up by my species, that's a measure of its dominance. Both of these are abundance measures. When I talk about species abundance, I could be talking about density or dominance, or this third one called frequency. Frequency is a measure of how common the species is within the community. Typically what you do is go out into the community, say that's my community, drop a few quadrats, say I had 5 quadrats, and I'll count the number of individuals of species 1 that I find. And let's say in 4 of the 5 quadrats I found species A, but I didn't find it in all of the quadrats. So this is a measure of presence divided by total number of quadrats looked at. Again, I'll lay these out a little bit more here in a bit, but for right now, the frequency of my species is 80% because it's present in 4 of the 5 quadrats. Doesn't matter how many quadrats or how many individuals are in each quadrat. It's really a measure of how common it is overall, presence, absence. Okay? All right. And then finally, this measure called species diversity. This is a measure of how kind of richness and abundance together form this idea of species diversity. So it's a metric that takes into account both abundance and richness. Okay, and again, we'll do these calculations later. I just want to present these to you initially, and then we're going to tear through each one. So the attributes that the community has, based on it being a closed community, that's the easiest way to do this, would be the number of species in the community, something about the abundance of those species as measured in 3 different ways, and some measure of the diversity of the community based on a combination of richness and abundance. Okay, so let's start out with species richness. Okay. So I've got species richness. I have a number of individuals within my species. So the first question I have for you is, how am I going to figure this out? Okay? Given that species are tough, right? They're tough to find. If I'm an animal biologist, for example, working in Presque Isle Bay, and I'm interested in the species richness of fish in Presque Isle Bay, I need to go out and sample them, and it probably matters how I sample them, right? I could go out and in my boat, right here, I could drag a trawl through the water. That's a net here. And I can collect the fish in this net, but the way the net's going through the water, I'm only collecting here the fish that are pelagic. I could get at that with something like a purse seine. Right, this is a net that samples fish up in the water column. There are also nets that can be dropped lower along the bottom because they have weights attached to them to hold them down. This would be something like an otter trawl. And depending on what type of net I use, I may get some types of species that tend to be pelagic but not benthic. And if I use this otter trawl, I may be collecting fish that are benthic but not pelagic. The size of the netting affects what kind of species I collect, right? If my mesh size is too large, I'm going to miss a lot of really tiny things, like minnows and maybe really, really tiny fish that can get through the net. Then I have the question of Presque Isle Bay is a very large kind of Presque Isle like this. Here, the bay itself is quite large. I have to make decisions about where to sample, right? Where to sample in the bay, what time of day, what time of year. Maybe some species are only resident, excuse me, at some times of the year, but not others. So I have some real decisions to make, and these decisions can affect my estimate of the species richness. If I'm out at the Erie Bluffs State Park, those of you not familiar with it, this is Route 5 right here. The Ohio border is like right over here. Girard is like right over there. Or sorry, not Girard. Fairview is like right here. Girard is more over there. Anyways, it's a state park. I think 2012 is when it officially became a state park. But if I were working in this state park and I were interested in animal species diversity, I have birds to collect. Well, in order to do that, how am I going to sample them? I could set up netting to catch them. I could sit out with a pair of binoculars. If I'm trying to collect small mammals, I've got maybe those small Sherman traps that I'm using to collect mice and voles and shrews, chipmunks. On the other hand, if I'm interested in deer and coyotes, maybe I've got traps that are just camera traps, right? Motion detection cameras take pictures of things. So just saying, well, I'm going to go out and figure out what the richness is of a particular community, it sounds very easy, but in actual practice, it's very hard. Different techniques are needed to figure out different types of species. Okay. If I'm interested in the insects out here, I'm probably going out with a net, an insect net, and trying to collect them all. Furthermore, you have a problem too in that a place like this, and even Presque Isle Bay, probably has a few different community types. And in fact, the Erie Bluffs State Park has about 12 different community types that have been classified. The bluffs themselves are close to the shoreline. That's an 80-foot drop, kind of straight down. The soil is very silty. That's considered to be a particular community type called an escarpment type. This area in here is a large sand dune. That's classified as a black oak savanna. Then you have other groups. This is an old hardwood, late successional hardwood forest here. On the other hand, these areas here are early successional, composed of different tree species. And based on the kinds of tree species that are there, like I said, there are 12 different community classifications here. So if I'm going to figure out how many species are out in this park, I have to have a pretty elaborate kind of sampling design to make sure I sample from each of these community types, since I recognize that different species tend to hang out in different community types. Okay, so let's narrow ourselves down to this small section of the park here. This is an abandoned field. And I thought what I would do is, instead of kind of talking about these things in abstract, is take a data set that I've got going back to, I think, I can't even remember what year it was, 2011 maybe? I had a student, she and I would go out to the bluffs every few days, and look at the species that occurred within this abandoned field. So the plan back in 2009, 2010 was to take the park, which had just been created, was going to be created in 2012, and the state was currently leasing out land for corn and soybean production. So the plan was to allow these fields to go fallow and then look at what species began to move into these communities over time. So a student and I looked at the first year following abandonment. I think that might have been 2009. Now I'm starting to wonder about myself here. Yeah, I think it was 2009, and then the work was published in 2011. So what we did was we went out into this park, into this abandoned field in 2009. Now it was abandoned in 2009 in the fall. That would have been when last planting. So what we did is go out in 2010, it's all coming back to me, 2010, and look at the species that occurred within that abandoned field. Okay? All right, this work has been published as the first year colonization of an old field within the Erie Bluffs State Park. This is a picture of that abandoned field in 2010. In 2009, this is just dirt. Right? At the end of 2009, the farmer collected all the corn, and there's nothing growing here. 2010, this is what it looks like. So what we did was we set up a sampling regime that we felt would increase the likelihood of us being able to pick out all of the species that occurred in the park, in the abandoned field. Okay? So what we did was we took this abandoned field here, so I'm just going to kind of draw this shape like this. That's the abandoned field. Okay, the abandoned field. And we set up transects. The transects are defined by the black arrows, and within each transect